Mr. Body here with our next biology lesson, this time on the cell cycle. And really what the cell cycle is, it's the lifespan that a typical cell will undergo. Now, all cells go through these three major phases, which are outlined in this diagram here, which are interphase, which you can see here, which spans the most of it, then mitosis and cytokinesis. Those are really our three phases of the cell cycle. Now, the frequency with how quickly these phases are completed are determined by the cell type. And we'll get into more of that later, but not every cell completes its cell cycle in the same amount of time frame. Like, for example, nerve cells take a really, really long time to go through this whole process, whereas skin cells go through it relatively quickly. So the type of cell will determine the frequency of it, but all cells have these same three major phases to their lifespan, which are interphase, mitosis, and cytokinesis. Interphase, as you can see, is broken up into three subphases, being G1, S, and G2. And you can see what's happening in each of those. In G1, the cell is growing. In G2, what's actually happening is the cell is doubling its DNA. It's making an exact copy of it in preparation for cell division. And then finally, G2 is just another form of cell growth. Then we get to mitosis, which is technically defined as the division of the nucleus itself. And then finally, cytokinesis, which is actual cell division. And so when we take a look at that, that's the whole process of the cell cycle. That's the whole cell life. Here's essentially all of the text from what I was just saying in terms of spelling out the phases: interphase, mitosis, cytokinesis, and then finally all of the subphases in mitosis and cytokinesis in there. Okay, so before we dive into looking at this process of the cell cycle in a little bit more detail, we need just a little bit of background knowledge on DNA. And so right here, what I have an image of, it's a chromosome, or oftentimes called a double chromosome, because it has two halves to it. A piece of DNA over here on the left-hand side, and a piece of DNA over here on the right-hand side, and they're held together by a structure in the middle. It's called a centromere. Now, just one of these pieces of DNA is called a chromatid. Now this chromatid here is exactly identical to this chromatid here, and these are the products of S phase. After S phase, we get this chromosome here and this chromosome here that are completely identical to each other because one of them is used as a template to make the other. Now, when the cell is not dividing though, it just has one copy of each chromosome in a different form of DNA. And that form of DNA is called chromatin, which is spelled out here. So when a cell is living its life, when it's in interphase, and interphase is by far the longest phase of the cell cycle, this is when a cell is just doing its job. It's doing the things that it needs to do to survive. So for example, in the human, if it's a skin cell, it's when it's being a skin cell. If it's a muscle cell, it's when it's being a muscle cell. Longest phase, broken into three subphases. During this phase, again, you can take a look, here's the term chromatin. Chromatin is a form of DNA which is really long and thin. Now, I say invisible here. What I mean by that is that it's not easily seen under a microscope. What's going to happen is that DNA is going to condense down into the chromatids or the chromosomes, and then they become visible under the microscope. In this form of DNA, in the form of chromatin, that DNA is usable. It can be easily read. Once it gets condensed down and coiled up, it's not so easily seen. Some other things to take note of inside of a cell that's in interphase is the nucleus and the nuclear membranes. The membrane that surrounds the nucleus is fully intact. And you have these structures that are called centrioles, which are going to be useful in the movement of chromosomes. They begin to move to opposite sides, or the poles of the cells. So one might move over here, and the other one might move over here, or top and bottom. Ultimately, it doesn't matter, as long as they separate itself away from either end of the cell. Then we start to get into the actual process of mitosis. And the first phase of mitosis is called prophase. And there's quite a bit that's happening here in prophase. In prophase, what we're seeing is that we're seeing the DNA condense down. Again, it's going from chromatin form into the chromatid or chromosome. Chromosomes and chromatid are kind of interchangeable words. And you can see them forming this characteristic X shape inside of the nucleus. And then the nuclear membrane itself, the membrane that surrounds the nucleus, it starts to break down and go away. And the reason for that is that it gives us access to these actual chromosomes so that they can be moved around. After the nuclear membrane's gone, we start to get these structures here that are called spindle fibers. A great analogy to these is that they're ropes. They're just ropes made of protein, and they come off of the centrioles. The centrioles are the structures that start to move to the poles. Then that whole structure put together, spindle fibers plus centrioles, it has its own name, it's called a centrosome. So those centrosomes are going to be responsible for the movement of the chromosomes eventually to opposite sides of the cell. So again, what we see happening in prophase, chromatin condense, nuclear membrane goes away, and we start to form these spindle fibers. The next phase is called metaphase, and really metaphase is an alignment phase, and all of the chromosomes here are going to be lined up down the middle or the equator of the cell. I always remember this as I remember metaphase, middle. They both start with M. So what's happening here is all of the DNA is stacked up and lined up right down the middle. The chromosomes have been attached to at the centromere by these spindle fibers. One spindle fiber on each side attaches to the centromere. Remember, those are the structures found right in the middle of the chromosomes. And again, an alignment thing. By having all of the chromosomes lined up down the middle here, we can account for all the chromosomes. And then eventually when they start to get pulled apart from each other, we can then ensure that equal amounts of chromosomes are pulled apart to opposite sides, which then of course takes us to anaphase, the third step of mitosis. Really what's happening here is that the chromosomes are being pulled apart. Remember, they're formed in that X, where the two halves of the chromosomes are exactly the same. Well, now they're being broken apart. The centromeres are broken, and each individual chromatid moves to opposite sides of the cell. This is the action phase of mitosis. This is where, again, the chromosomes are physically pulled apart into two separate sides, and that's all coordinated by the centrosomes in combination with the spindle fibers. Then we have telophase. Telophase is pretty much the exact opposite of prophase. So in telophase, we're going to uncoil those chromatids, turn them back into the usable form of chromatin. We get a nuclear membrane forming around each set of chromosomes. So there's a new nuclear membrane here and a new nuclear membrane here. The spindle fibers are no longer needed, so they break down. And what will actually start to happen is the cell will start to pinch together. You can see the cell membrane coming together here and down here as well. The cell is starting to pinch in preparation for cell division. But at the end of telophase now, you have one cell and there's two nuclei inside of it. And so essentially now mitosis, which is nucleus division, is done. The nucleus is such an important organelle that it gets its own phase because the DNA is ultimately the stuff that controls all the functions of the cell. So mitosis is a phase that's just used for nucleus division. Finally, then we have cytokinesis, and this is cell division. It's also categorized as cytoplasm division. But now the cell here will fully split in half, and now you end up with two daughter cells that are completely identical to each other and to the original starting cell. These two cells now are free to go back into interphase and to do their job. So cytokinesis is kind of the ending phase now. Now you have two cells that are smaller than the original, and they'll go through the growth process with an interphase. They'll go through S phase again, they'll go through G2, and then eventually they'll go through the mitosis phase, followed up by cytokinesis. So that's a quick look at the whole process of the cell cycle, starting with interphase and working our way all the way through mitosis, and then finally cytokinesis.